Welcome to our podcast on hydrodynamic focusing for single-cell analysis on a chip. Today, we begin an extensive discussion into how precise microfluidic techniques are revolutionizing the way we analyze individual cells. This technology leverages controlled microflows to align cells into a narrow stream on a chip, thereby enabling high-resolution single-cell sequencing and opening new avenues in personalized medicine. We'll explore the underlying physics, innovative channel design, engineering challenges, and practical applications that together promise to transform cellular analysis.
Thank you for having me. I'm delighted to join this conversation and share insights on how hydrodynamic focusing is advancing single-cell analysis. This field is truly exciting as it marries fluid mechanics, microfabrication, and cell biology to achieve precise control over cell positioning. Such precision is essential for capturing high-quality data from single cells, which is the cornerstone of personalized diagnostics and targeted therapies.
Let's start with the basics. Could you explain what hydrodynamic focusing is and why it is critical for single-cell analysis on a chip? I'd like to understand the fundamental principles that make this technique so powerful.
Certainly. Hydrodynamic focusing is a microfluidic technique that uses carefully controlled fluid flows to confine a sample stream typically containing cells into a very narrow, well-defined core. In practice, this is achieved by introducing one or more sheath flows alongside the central sample flow. Because the flows are laminar in microchannels, they do not mix turbulently but instead remain as parallel streams. This pinching action compresses the sample stream, ensuring that the cells are aligned in a single file along the center of the channel. This precise alignment is essential for downstream processes such as optical detection, droplet encapsulation, or electrical impedance measurements, as it guarantees that each cell passes through the detection region individually and predictably.
That makes sense. I understand that fluid dynamics in microchannels plays a major role here. Could you elaborate on how the low Reynolds number in these systems facilitates hydrodynamic focusing and what implications this has for the design of the microchannels?
Absolutely. In microfluidic systems, the Reynolds number, a dimensionless parameter that indicates the ratio of inertial forces to viscous forces, is typically very low, meaning that viscous forces dominate. This ensures laminar flow, where fluid moves in parallel layers with minimal mixing due to turbulence. Because of this predictable, orderly flow, when you inject a central sample flow and flank it with sheath flows, the viscous forces from the sheath fluid steadily compress the sample stream into a narrow, focused core. This predictable behavior allows engineers to design microchannels with specific geometries that precisely control the width of the focus stream. The design parameters, including channel dimensions and flow rates, are often optimized using computational fluid dynamics (CFD) simulations, which help predict how the fluids and cells will behave within the channel.
That's very informative. Now, I'd like to move on to the practical challenges. When dealing with biological samples, cells vary widely in size, shape, and mechanical properties. How does hydrodynamic focusing accommodate such heterogeneity? And what design strategies are being developed to optimize cell alignment for different cell types?
Excellent question. One of the key challenges in hydrodynamic focusing is indeed dealing with cellular heterogeneity. Cells can vary not only in size, but also in deformability, which affects how they interact with fluid flows. To address this, researchers are developing adaptive channel designs that can accommodate a range of cell sizes. For example, some designs incorporate tapered channels where the width gradually decreases, gently guiding cells into alignment. Others employ channel geometries with curved or serpentine sections that generate secondary flows to help center cells, regardless of their size or shape. Additionally, dynamic flow control systems are being developed that adjust the ratio of sheath to sample flow in real time based on feedback from integrated sensors. These adaptive systems ensure that even if the sample contains a mix of cell types, the focusing remains precise and consistent.
That leads me to another concern. Some scientists have debated whether the forces required for tight focusing might induce shear stress that could harm the cells. What evidence do you have that hydrodynamic focusing can be achieved without compromising cell viability, and how are researchers balancing alignment precision with cell health?
Shear stress is indeed a valid concern in microfluidic systems. High shear forces can damage cells, which would be detrimental, especially in applications like single-cell sequencing where cell integrity is paramount. Researchers have addressed this by carefully optimizing the flow rates and channel dimensions to minimize shear stress. For instance, by adjusting the viscosity of the sheath fluid or by incorporating channel designs that gradually compress the sample stream, the shear forces acting on the cells can be reduced. Numerous viability assays and functional tests have confirmed that when these parameters are optimized, cells remain healthy and fully functional after passing through the focusing region. In fact, many studies report high viability rates, sometimes exceeding 90%, which indicates that the technique is gentle enough for sensitive biological applications.
Look, let's talk about channel design in more detail. Could you explain how the geometry of the microchannels affects cell alignment and what specific design elements have been shown to improve the precision of hydrodynamic focusing?
Certainly, the geometry of the microchannels is critical for achieving optimal cell alignment. In its simplest form, a straight channel with constant width can provide basic focusing. However, advanced designs incorporate features that enhance the uniformity of the focused stream. For example, serpentine channels create a series of turns that help to center the cells by repeatedly altering the direction of the flow, which minimizes any lateral deviations. Staggered herringbone mixers are another innovation. These are patterned on the channel walls to induce microvortices that promote mixing and help guide cells into the center of the channel. Some designs even incorporate three-dimensional structures that allow for more complex flow patterns, further improving alignment precision. Researchers use both experimental studies and CFD simulations to test various geometries and determine the optimal design for specific applications. The goal is to achieve a narrow, stable, and reproducible cell stream that can be reliably used for downstream analysis.
I'd like to shift our discussion to the fluids used in these systems. How do the properties of the sample and sheath fluids influence the focusing process, and what considerations must be taken into account when selecting these fluids for biological applications?
Fluid properties are indeed crucial. The sample fluid, which contains the cells, is usually a biological medium, while the sheath fluid is a carefully formulated buffer. Key properties such as viscosity, density, and surface tension determine how the fluids interact in the microchannel. A higher viscosity in the sheath fluid can enhance the focusing effect by exerting a stronger compressive force on the sample stream. However, the viscosity must be balanced so as not to introduce excessive resistance that could lead to pressure buildup or unstable flows. Additionally, the density and surface tension of the fluids affect the interface dynamics between the sample and sheath streams. It is essential that the fluids are compatible to prevent any adverse effects on the cells, such as osmotic shock or chemical incompatibility. Researchers often conduct systematic studies to determine the optimal fluid compositions that provide effective focusing while preserving cell viability and functionality.
Now, let's delve into the implications for single-cell sequencing. How does hydrodynamic focusing improve the accuracy and efficiency of single-cell sequencing? And can you provide specific examples of its impact on the quality of sequencing data?
Hydrodynamic focusing has a profound impact on single-cell sequencing by ensuring that individual cells are isolated and aligned in a consistent manner. In single-cell sequencing, it is imperative that each cell is processed independently to avoid cross-contamination and to ensure that the genetic material from one cell is not mixed with that of another. When cells are tightly aligned in a narrow stream, they can be accurately encapsulated into droplets or sorted for analysis. This precise control over cell positioning minimizes variability in the data and results in higher quality sequencing outputs. For instance, studies have shown that platforms incorporating hydrodynamic focusing yield more uniform sequencing coverage and a lower rate of doublets, instances where two cells are inadvertently processed together, which significantly improves the reliability of gene expression data. This level of precision is particularly critical in applications such as cancer research, where identifying rare cell subpopulations can lead to important insights into tumor heterogeneity and treatment resistance.
Personalized medicine is one of the most exciting applications of single-cell analysis. Could you discuss how advances in hydrodynamic focusing are contributing to personalized medicine, particularly in terms of tailoring treatments based on individual cellular profiles?
Personalized medicine relies on detailed molecular information from individual cells to tailor treatments to each patient's unique genetic makeup. Hydrodynamic focusing plays a vital role by enabling the precise isolation of single cells, which is the first step in high-resolution analysis. When each cell can be individually sequenced and characterized, clinicians gain insights into the heterogeneity within tissues, such as tumors. This detailed information allows for the identification of specific genetic mutations, differential gene expression patterns, and rare cell types that may influence how a patient responds to a particular therapy. For example, in oncology, hydrodynamic focusing has been used to isolate circulating tumor cells from blood samples, providing a non-invasive means of monitoring tumor evolution and treatment response. Such data can inform the design of targeted therapies that address the specific molecular drivers of an individual's cancer, ultimately improving treatment efficacy and reducing side effects. The ability to perform such personalized analyses is a major step forward in the era of precision medicine.
Some researchers have debated the trade-offs between throughput and accuracy in hydrodynamic focusing systems. How do you reconcile the need for high throughput analysis with the requirement for precise cell alignment, and what strategies are being developed to address this balance?
This is a topic of active research and sometimes debate. On one hand, high throughput analysis is essential for processing large numbers of cells quickly, which is critical for applications like population-scale studies or clinical diagnostics. On the other hand, precise cell alignment is necessary to ensure that the data collected from each cell is accurate and reliable. Balancing these requirements involves optimizing channel design, flow parameters, and detection systems to achieve both high throughput and high precision. Researchers are exploring dynamic flow control systems that can adjust in real time based on feedback from integrated sensors. For instance, if a sensor detects slight deviations in cell alignment, the system can immediately modify the flow rates to re-establish optimal conditions. Additionally, modular designs allow for parallel processing where multiple microchannels operate simultaneously, effectively increasing throughput without compromising the precision of each individual channel. This integrated approach has shown promising results, with studies reporting that high-throughput systems can achieve near-perfect cell alignment while maintaining rapid processing speeds.
Another aspect worth discussing is the integration of on-chip sensors for real-time monitoring of cell alignment and viability. Can you elaborate on the types of sensors being integrated and how they contribute to the overall performance and reliability of the system?
Certainly. Integrating on-chip sensors into microfluidic devices has become a pivotal part of optimizing hydrodynamic focusing. Optical sensors, for example, are widely used to capture high-resolution images of the cells as they pass through the channel. These sensors can detect variations in cell size, shape, and fluorescent signals, providing immediate feedback on alignment quality and cell health. In addition to optical methods, electrical impedance sensors are being incorporated to measure changes in the electrical properties of cells. These sensors can detect differences in cell membrane integrity and cytoplasmic conductivity, which are indicative of cell viability. Some platforms also employ mechanical sensors that measure pressure and flow dynamics within the channel. The data from these various sensors is processed in real time by integrated microprocessors, which use adaptive algorithms to fine-tune the flow conditions. This continuous monitoring ensures that any deviations from optimal focusing are corrected immediately, enhancing both the accuracy and reproducibility of single-cell analysis.
While these technological advancements are promising, they also raise important questions about the potential impact of hydrodynamic focusing on cell physiology. There is ongoing debate about whether the forces exerted during focusing could alter cellular behavior or gene expression. What is your perspective on this issue, and what evidence supports the safety of these techniques for downstream biological analysis?
This is an important concern, and it has been the subject of extensive investigation. While it is true that the forces exerted on cells during hydrodynamic focusing can, in theory, influence cellular behavior, empirical evidence suggests that when flow parameters are carefully optimized, the impact on cell physiology is minimal. Numerous studies have employed viability assays and gene expression analyses to assess the effect of hydrodynamic focusing on cells. These studies have generally found that under optimized conditions, the process does not significantly alter cell viability or trigger stress responses that could skew downstream analyses. In fact, the gentle, controlled nature of the microfluidic environment is often cited as an advantage over more invasive cell sorting methods. That said, it remains critical for each application to carefully calibrate the system to ensure that cells remain as close to their native state as possible, particularly when sensitive downstream analyses such as single-cell sequencing are involved.
Let's now turn to the topic of materials and fabrication. Can you provide an overview of the fabrication process for hydrodynamic focusing chips used in single-cell analysis? Specifically, what materials and techniques are most commonly employed, and how do they contribute to the performance and reliability of the device?
Hydrodynamic focusing chips are typically fabricated using microfabrication techniques borrowed from the semiconductor industry. Commonly, polymers such as PDMS, polydimethylsiloxane, and COC, cyclic olefin copolymer, are used to create the microchannels. These materials are favored for their biocompatibility, optical transparency, and ease of fabrication via soft lithography or injection molding. The fabrication process generally involves creating a master mold using photolithography, from which the microchannel structures are replicated in PDMS. Once the channels are formed, they are bonded to a glass or plastic substrate, which provides structural support. In some cases, advanced techniques like laser micromachining or 3D printing are employed to create more complex channel architectures. The choice of materials and precise fabrication methods are critical to ensuring that the channels are smooth and reproducible, which in turn is essential for achieving consistent hydrodynamic focusing. Furthermore, surface treatments and coatings may be applied to the channel walls to prevent nonspecific cell adhesion and to improve fluid dynamics, thus enhancing the overall performance of the device.
Data management and real-time analysis are also integral to modern hydrodynamic focusing systems. How do you envision the integration of data analytics with these microfluidic platforms, and what role does artificial intelligence play in optimizing cell alignment?
Data analytics and artificial intelligence are rapidly transforming the field of microfluidics. With the integration of high-precision sensors and real-time imaging, hydrodynamic focusing systems generate enormous amounts of data during cell processing. Advanced algorithms, particularly those based on machine learning, are being developed to analyze this data in real time. These AI systems can detect subtle deviations in cell alignment, predict when a channel might become clogged, and automatically adjust flow parameters to maintain optimal focusing. For instance, by analyzing changes in the position and velocity of cells as they pass through the channel, the system can determine if the focusing is suboptimal and then modulate the sheath-to-sample flow ratio accordingly. This level of automation not only increases the throughput of single-cell analysis, but also ensures that the quality of the data remains high, thereby improving the reliability of downstream applications such as sequencing and diagnostics.
Moving forward, what do you see as the most critical research challenges in the field of hydrodynamic focusing for single-cell analysis? Are there any debates or controversies among researchers regarding the best approaches to optimizing cell alignment?
One of the primary challenges is achieving consistent cell alignment across a wide range of cell types and conditions. Because cells vary in size, shape, and mechanical properties, a channel design that works well for one cell type might not be optimal for another. This variability has led to active debates among researchers about whether a standardized channel design can be developed, or if systems need to be customized for each specific application. Another challenge involves balancing the precision of cell alignment with the need to maintain high cell viability. Some researchers argue that tighter focusing could inadvertently subject cells to higher shear stress, potentially affecting their functionality, While many studies have demonstrated that careful optimization can mitigate these effects, the issue remains a topic of discussion and investigation. Additionally, integrating multimodal detection systems into a single compact chip poses significant engineering challenges that continue to drive research efforts in this field.
Let's explore the clinical applications of hydrodynamic focusing. In what ways is this technology advancing single-cell sequencing and personalized medicine? And could you provide some concrete examples of its impact on clinical diagnostics or treatment strategies?
Hydrodynamic focusing is a game changer for single-cell sequencing because it enables the precise isolation and alignment of individual cells, which is crucial for generating high-quality sequencing data. In clinical diagnostics, for instance, this technology allows for the detection of rare cell populations such as circulating tumor cells, which can provide early indications of cancer. One study demonstrated that by using hydrodynamic focusing to isolate single cells from a patient's blood sample, researchers were able to perform high-resolution sequencing that identified genetic mutations associated with aggressive tumor behavior. This kind of data is invaluable for designing targeted therapies that are tailored to the genetic profile of a patient's tumor, thus advancing the field of personalized medicine. Similarly, in immunology, hydrodynamic focusing has been used to isolate individual immune cells for detailed analysis, which can help in understanding immune responses and in developing personalized immunotherapies.
On a related note, how do you address the potential ethical concerns associated with single-cell analysis in a clinical setting, particularly regarding data privacy and the potential misuse of genetic information?
Ethical considerations are paramount in any technology that deals with sensitive biological data. In the context of single-cell analysis, strict protocols must be established to ensure data security and patient privacy. This includes the use of robust encryption methods, secure data storage, and strict access controls to ensure that genetic information is only used for its intended purpose. Regulatory frameworks also play a critical role in governing how such data is collected, shared, and used. It is essential that researchers and clinicians work closely with ethicists and legal experts to develop guidelines that protect patient rights while still enabling scientific and clinical advancement. Transparency with patients regarding the use of their data, informed consent, and ongoing ethical oversight are all vital components of responsibly implementing this technology in personalized medicine.
Let's shift focus to the integration of hydrodynamic focusing with other microfluidic technologies. How are researchers combining hydrodynamic focusing with cell sorting, lysis, and molecular analysis on a single chip to create a fully integrated lab-on-a-chip platform?
One of the most exciting developments in this field is the emergence of fully integrated lab-on-a-chip devices that combine hydrodynamic focusing with other critical processing steps, such as cell sorting, lysis, and nucleic acid extraction. In these systems, hydrodynamic focusing is used as the initial step to align and isolate individual cells. Once focused, the cells can be directed into downstream modules where they undergo lysis to release their intracellular contents. Subsequent modules then perform nucleic acid extraction, amplification, and even preliminary sequencing. This integration minimizes manual handling, reduces the risk of contamination, and significantly speeds up the workflow, making it possible to perform complex single-cell analyses in a matter of minutes. Such devices are particularly promising for point-of-care diagnostics, where rapid and accurate cellular analysis can have a profound impact on patient care.
Another key area of research involves the development of new channel materials and surface coatings to enhance the performance of hydrodynamic focusing devices. Could you discuss how innovations in material science are contributing to the reliability and efficiency of these platforms?
Material science plays a crucial role in the advancement of microfluidic devices. Researchers are actively exploring new polymers and surface modification techniques to improve the performance of hydrodynamic focusing chips. For instance, developing surface coatings that minimize cell adhesion is vital for preventing clogging and ensuring a smooth, continuous flow of cells through the channels. Innovations in this area include the use of anti-fouling coatings in bioinert polymers that reduce nonspecific binding of cells to the channel walls. In addition, advancements in the fabrication of microchannels, such as improved soft lithography techniques and high-resolution 3D printing, have enabled the production of channels with highly consistent dimensions and smooth surfaces, which are essential for achieving precise cell alignment. These material innovations not only enhance device performance, but also contribute to the overall durability and scalability of the technology.
Debates in the research community often focus on the optimal balance between throughput and precision in hydrodynamic focusing systems. How do you see this balance evolving, and what strategies are being employed to ensure that high-throughput systems do not compromise on the accuracy of single-cell analysis?
This balance is indeed one of the central challenges in the field. High-throughput analysis is critical for applications such as large-scale genomic studies and clinical diagnostics, but it must not come at the expense of precision. To address this, researchers are developing parallelized microfluidic systems that process multiple streams simultaneously. By designing chips with an array of identical microchannels operating in parallel, it is possible to achieve high throughput while maintaining the stringent focusing conditions required for precise single-cell analysis. Additionally, advanced control systems that incorporate real-time feedback from integrated sensors are being used to continuously monitor and adjust flow conditions, ensuring that the alignment remains consistent across all channels. This combination of parallel processing and adaptive control represents a promising strategy for achieving both high throughput and high precision.
Let's consider the role of computational modeling in advancing hydrodynamic focusing. How are computational fluid dynamics, CFD, simulations being used to design and optimize microchannels for single-cell analysis?
Computational fluid dynamics, CFD, is a powerful tool that allows researchers to simulate and analyze fluid behavior at the microscale. CFD models are used to predict how cells will behave under different flow conditions and channel geometries, which is critical for optimizing the design of hydrodynamic focusing devices. By running simulations, engineers can identify the optimal flow rates, channel dimensions, and design features that will result in the best cell alignment. These simulations enable rapid prototyping and iterative design improvements without the need for extensive physical testing, which saves both time and resources. CFD modeling has been instrumental in advancing our understanding of the fluid dynamics within microchannels and is a key factor in the successful development of high-performance hydrodynamic focusing platforms.
Now, let's delve into some of the economic and regulatory aspects. What are the key challenges in commercializing hydrodynamic focusing technology for single-cell analysis, and how are researchers addressing these challenges to make the technology accessible for clinical and research applications?
Commercializing hydrodynamic focusing technology involves overcoming several hurdles. One major challenge is ensuring reproducibility and scalability. Laboratory prototypes often perform exceptionally well under controlled conditions, but translating these results into mass-produced devices requires rigorous standardization of fabrication processes and materials. Researchers are collaborating with industry partners to develop scalable manufacturing techniques such as injection molding and roll-to-roll processing, which can produce high-quality chips at a lower cost. On the regulatory front, establishing clear standards for device performance, safety, and data reliability is critical. Regulatory agencies are beginning to work with academic and industrial stakeholders to develop testing protocols and certification processes tailored to microfluidic single-cell analysis platforms. These collaborative efforts aim to create a supportive regulatory environment that fosters innovation while ensuring that the technology meets the high standards required for clinical applications.
Turning to education, how are academic institutions preparing the next generation of scientists and engineers for work in this interdisciplinary field? What changes in curricula and training programs are needed to support the rapid advancement of hydrodynamic focusing technologies?
Academic institutions are increasingly recognizing the need for interdisciplinary training to advance microfluidic technologies. Curricula are being updated to integrate courses in fluid mechanics, material science, bioengineering, and data analytics, providing students with a well-rounded foundation in both theory and practical applications. Many universities have established interdisciplinary research centers and labs where students from diverse fields collaborate on projects related to single-cell analysis and microfluidics. Hands-on training through laboratory work, internships, and cooperative education programs with industry partners is also essential for equipping students with the practical skills needed to innovate in this field. By fostering an environment of cross-disciplinary collaboration and continuous learning, academic institutions are preparing the next generation of researchers to drive future breakthroughs in hydrodynamic focusing and its applications in personalized medicine.
Looking toward the future, what are some of the most exciting research directions or breakthrough applications you foresee in the field of hydrodynamic focusing for single-cell analysis? How might these innovations reshape our approach to diagnostics and personalized therapy?
The future of hydrodynamic focusing for single-cell analysis is full of exciting possibilities. One particularly promising direction is the development of fully integrated lab-on-a-chip platforms that combine cell alignment, sorting, lysis, and molecular analysis into a single automated device. Such platforms could enable rapid point-of-care diagnostics by allowing clinicians to analyze a patient's cells in real time and make immediate data-driven treatment decisions. In personalized medicine, the ability to obtain high-resolution single-cell profiles will be critical for tailoring therapies to the unique genetic and molecular characteristics of an individual's cells. For example, in cancer treatment, detailed analysis of individual tumor cells can reveal the presence of rare, drug-resistant subpopulations, thereby informing more effective targeted therapies. Additionally, advances in hydrodynamic focusing could drive innovations in high-throughput drug screening, where rapid analysis of cellular responses to different compounds accelerates the discovery of new therapeutic agents.
Another visionary application is the potential for hydrodynamic focusing to enhance the field of immunotherapy. The immune system is highly complex and heterogeneous, and understanding its nuances at the single-cell level is essential for developing effective treatments. By precisely aligning immune cells on a chip, researchers can study their responses to various stimuli, identify rare cell types, and unravel the complex signaling networks that govern immune responses. This level of analysis could lead to the development of highly personalized immunotherapies that are tailored to the specific immune profiles of patients, potentially revolutionizing the treatment of autoimmune diseases and cancer.
Furthermore, the integration of hydrodynamic focusing with emerging digital technologies is set to create new research paradigms. The convergence of microfluidics with big data analytics and artificial intelligence is giving rise to energy informatics for single-cell analysis, where sophisticated algorithms process vast datasets to optimize device performance and predict cellular behavior. This interdisciplinary fusion not only improves the efficiency of single-cell analysis, but also opens up novel avenues for understanding complex biological systems. For instance, AI-driven models can analyze single-cell sequencing data to identify previously unknown cell subtypes or predict responses to therapeutic interventions, thereby enhancing our understanding of disease mechanisms and informing the development of new treatment strategies.
Lastly, as these technologies mature, there will be important considerations regarding their global impact. In regions with limited access to advanced diagnostic tools, portable, cost-effective, single-cell analysis platforms based on hydrodynamic focusing could transform healthcare delivery. Decentralized diagnostic systems that are capable of rapid, high-resolution cellular analysis would enable early detection of diseases and prompt intervention, ultimately improving patient outcomes in underserved communities. Moreover, the scalability and low resource requirements of microfluidic devices make them an attractive option for reducing healthcare disparities worldwide.
In summary, the advanced integration of hydrodynamic focusing with state-of-the-art detection, control, and data analytics systems is paving the way for a new era in single-cell analysis. This technology has the potential to revolutionize personalized medicine, accelerate drug discovery, and drive transformative changes in both research and clinical practice. As interdisciplinary collaboration continues to flourish, we can expect further breakthroughs that will refine cell alignment technique, improve device scalability, and expand the applications of this powerful technology.
Thank you for engaging in this detailed exploration of advanced integration, regulatory and economic considerations, and future research directions for hydrodynamic focusing for single-cell analysis on a chip. I hope that our discussion has provided you with deep insights into the current challenges and emerging opportunities in this field. In our next segment, we will delve even further into cutting-edge sensor technologies, adaptive control strategy, and the visionary potential of these devices in personalized medicine and beyond.
This concludes our extended discussion on advanced topics in hydrodynamic focusing for single-cell analysis. Thank you for listening, and I look forward to our continued conversation as we explore new horizons in microfluidic technology and its transformative impact on healthcare research and environmental monitoring. Stay curious, stay engaged, and keep pushing the boundaries of what is possible.
Welcome back to our discussion on hydrodynamic focusing for single-cell analysis on a chip. Today, we're shifting our focus toward advanced integration techniques and exploring the broader implications of this technology for next-generation diagnostics and personalized medicine. Let's dive right in.
Thank you for having me again. I'm excited to discuss how we're moving beyond the basic principles of cell alignment and into the realm of integrated, multifunctional, lab-on-a-chip platforms that are poised to revolutionize single-cell analysis and clinical diagnostic.
To begin, can you explain how researchers are integrating hydrodynamic focusing with downstream detection systems? What are the new sensor technologies being embedded on the chip, and how do they improve the real-time monitoring of individual cells?
Certainly. In advanced hydrodynamic focusing platforms, we're no longer just aligning cells, we're coupling this process with a suite of integrated sensors. For example, many researchers now embed high-speed optical sensors such as miniature fluorescence microscopes directly into the microfluidic chip. These sensors capture real-time images of cells as they flow through the focus stream, enabling immediate analysis of cell morphology and fluorescence markers. Electrical impedance sensors are integrated to monitor cell membrane integrity and detect subtle changes in cellular electrical properties. In some designs, mechanical sensors are also used to measure pressure variations within the channels. The real-time data collected by these sensors is processed on chip by microprocessors, which feed information into adaptive control algorithms. This integration allows for continuous adjustments to flow parameters, ensuring that cells remain optimally aligned. The result is a smart self-correcting system that enhances both the accuracy and throughput of single-cell analysis.
That's fascinating. It seems that combining different sensing modalities is key to building a robust platform. Can you discuss an example where this multimodal approach has led to significant improvements in cell analysis performance?
Absolutely. In one recent study, a research team developed a microfluidic chip that integrated both optical and impedance sensors. The optical sensors provided high-resolution images of cells, while the impedance sensors measured real-time electrical properties. By correlating the visual data with the impedance signals, the system was able to detect even subtle variations in cell size and membrane integrity. This dual modality approach allowed the researchers to not only ensure precise cell alignment, but also to identify cells that were under stress or in the process of apoptosis. Such detailed multi-emperament data proved invaluable in refining downstream single-cell sequencing protocols, ultimately resulting in higher fidelity genetic information. This example illustrates how integrating complementary sensor technologies can significantly enhance the overall performance and reliability of single-cell analysis platforms.
Moving beyond detection, let's talk about adaptive control systems. How are these systems using real-time data to optimize hydrodynamic focusing, and what challenges remain in implementing such feedback mechanisms?
Adaptive control systems are at the heart of modern microfluidic platforms. Essentially, these systems rely on real-time feedback from the integrated sensors to adjust the flow conditions dynamically. For instance, if the optical sensors detect that cells are starting to drift away from the center of the channel, the control system can instantly adjust the sheath-to-sample flow ratio to re-establish the optimal focusing conditions. One challenge in implementing these feedback loops is ensuring that the response time of the control system is fast enough to correct any deviations before they impact the quality of cell alignment. Moreover, the algorithms that govern these adjustments need to be robust and capable of handling variability between different cell types and sample conditions. Researchers are continuously refining these algorithms, often using machine learning techniques to predict and preempt potential alignment issues. While the progress so far is impressive, achieving seamless real-time control in a variety of experimental conditions remains an active area of research.
On a related note, computational fluid dynamics, CFD, plays a crucial role in designing these systems. Can you elaborate on how CFD simulations are used to optimize microchannel geometries and flow conditions for hydrodynamic focusing?
Certainly. CFD simulations are an indispensable tool for understanding fluid behavior in microchannels. By creating detailed computational models of the microfluidic system, researchers can simulate how different channel geometries and flow rates affect the behavior of cells. These simulations allow us to predict the impact of various design parameters such as channel width, curvature, and the ratio of sheath to sample flow. CFD helps identify regions of recirculation or high shear stress, which are critical for ensuring cell viability and precise alignment. By iteratively refining the design in silico, we can optimize the channel architecture before fabricating physical prototypes. This not only saves time and resources, but also leads to more efficient and reliable devices. In many cases, CFD simulations have revealed subtle design improvements that significantly enhance cell focusing, underscoring the value of this approach in developing advanced microfluidic platforms.
Let's turn our attention to regulatory challenges. As these technologies mature and move closer to clinical application, what regulatory hurdles must be overcome, and how are researchers working with regulatory bodies to establish standards for hydrodynamic focusing devices?
Regulatory challenges are indeed a major consideration as we transition from the lab to clinical use. Microfluidic devices for single-cell analysis operate at a scale and under conditions that differ significantly from conventional diagnostic tools, which means that existing regulatory frameworks often need to be adapted. Researchers are working closely with regulatory agencies to develop new standards that address key issues such as device reproducibility, cell viability, and data accuracy. This collaborative process involves establishing rigorous testing protocols, calibrating sensor outputs, and defining performance benchmarks that these devices must meet. In some cases, early pilot studies are conducted under regulatory oversight to generate the necessary data for approval. The goal is to create a clear, supportive regulatory pathway that not only ensures patient safety but also encourages innovation by providing predictable standards for device performance. This collaboration between researchers, manufacturers, and regulators is essential for building public trust and facilitating the widespread adoption of these technologies in clinical settings.
Economically, what factors are driving the adoption of hydrodynamic focusing technologies? And how do you see the cost-benefit balance evolving as these devices scale up?
From an economic perspective, the adoption of hydrodynamic focusing technologies is driven by several key factors. Although the initial costs of development and fabrication can be relatively high due to the advanced materials and precision engineering required, the long-term benefits are substantial. High throughput, single-cell analysis enabled by these devices can significantly reduce the time and expense associated with traditional diagnostic methods. Moreover, the increased accuracy and resolution of single-cell data have the potential to improve treatment outcomes in personalized medicine, thereby reducing overall healthcare costs. As manufacturing processes mature and economies of scale are realized, the per-unit cost of these devices is expected to decline dramatically. Additionally, the environmental benefits such as reduced waste and lower energy consumption translate into indirect economic gains by lowering the societal costs associated with healthcare and environmental remediation. Overall, the cost-benefit balance is expected to become increasingly favorable, making these technologies a compelling investment for both the private and public sectors.
How do you view the role of interdisciplinary collaboration in advancing hydrodynamic focusing technology And what initiatives or research programs are in place to foster such collaboration?
Interdisciplinary collaboration is absolutely critical to advancing hydrodynamic focusing technology. The field sits at the intersection of microfluidics, cell biology, materials science, computational modeling, and clinical research. Successful innovation in this area requires the combined expertise of engineers, biologists, chemists, data scientists, and clinicians. Many research institutions have established dedicated interdisciplinary centers focused on lab-on-a-chip technologies, where experts from different fields work together on projects aimed at optimizing cell alignment and integrating advanced detection systems. Collaborative grant programs and international research consortia also play a key role in fostering this kind of cross-disciplinary work. These initiatives encourage the exchange of ideas and techniques, leading to breakthroughs that would not be possible within the confines of a single discipline. The synergy generated by these collaborations is driving rapid progress in the field and paving the way for real-world applications in personalized medicine and beyond.
Let's discuss the future directions of hydrodynamic focusing technology. What do you envision as the next major breakthroughs or applications that could transform single-cell analysis and personalized medicine in the coming years?
Looking to the future, one of the most exciting prospects is the development of fully integrated, portable, lab-on-a-chip devices that combine hydrodynamic focusing with multimodal detection and on-chip molecular processing. Imagine a compact device capable of isolating single cells, lysing them, extracting nucleic acids, and performing preliminary sequencing, all within a matter of minutes. Such devices would revolutionize point-of-care diagnostics by providing rapid, high-resolution data that clinicians could use to make real-time treatment decisions. In personalized medicine, this could mean the ability to quickly generate comprehensive cellular profiles from a patient's blood sample, leading to more tailored and effective therapies. Another breakthrough on the horizon is the use of artificial intelligence to optimize and control microfluidic flows. With AI-driven systems, devices could automatically adjust flow rates and channel conditions based on real-time feedback, ensuring optimal cell alignment under any condition. This would not only improve data quality, but also increase throughput and reproducibility, making single-cell analysis more robust and scalable for large-scale clinical studies.
Another visionary application is in the field of precision oncology. As we improve our ability to analyze individual tumor cells, hydrodynamic focusing could be instrumental in uncovering the genetic diversity within cancers. This detailed cellular information would allow oncologists to identify subpopulations of tumor cells that are resistant to current treatments, leading to more targeted and effective therapeutic strategies. Such advancements could significantly improve patient outcomes and reduce the side effects associated with generalized chemotherapy regimens. Moreover, the insights gained from single-cell analysis could also facilitate the development of novel immunotherapies as researchers identify specific cell markers that can be targeted by new drugs.
On a broader scale, integrating hydrodynamic focusing with advanced microfluidic platforms holds promise for transforming not just biomedical research, but also environmental monitoring and industrial biotechnology. For example, in environmental applications, single-cell analysis of microbial communities can provide critical insights into ecosystem health and pollutant degradation. By precisely isolating individual microorganisms, researchers can study their metabolic profiles and interactions, leading to improved strategies for bioremediation and sustainable resource management. In industrial biotechnology, the ability to analyze individual cells in fermentation processes could lead to optimized production methods for biofuels and other bioproducts. These applications demonstrate the wide-ranging impact of hydrodynamic focusing technology beyond the realm of healthcare.
Considering the rapid pace of technological advancement, what are some of the emerging challenges that you foresee, and how might the research community address them in the coming years?
While the progress in hydrodynamic focusing has been remarkable, several challenges remain. One of the most pressing issues is achieving consistent cell alignment across diverse cell types, particularly when dealing with highly heterogeneous populations. Variability in cell size, shape, and mechanical properties can lead to discrepancies in focusing performance. To address this, the research community is exploring adaptive channel designs and dynamic flow control systems that can adjust in real time based on feedback from integrated sensors. Another challenge is the integration of multimodal detection systems without compromising the microfluidic flow. As we add more sensing components to the chip, ensuring that they do not interfere with the fluid dynamics becomes critical. This requires innovative design solutions and careful optimization of both the physical layout and the electronic integration. Additionally, as the technology moves toward clinical application, scalability and reproducibility will be paramount. Researchers must develop standardized fabrication protocols and robust quality control measures to ensure that devices perform reliably in diverse settings. Addressing these challenges will require sustained interdisciplinary collaboration and investment in advanced fabrication and control technologies.
How do you envision the role of data analytics evolving in this field, particularly in terms of handling the large data sets generated by high-throughput single-cell analysis?
Data analytics is becoming increasingly central to the field of single-cell analysis, as hydrodynamic focusing systems generate vast amounts of real-time data on cell behavior. Advanced analytics tools, especially those based on machine learning, are essential for processing and interpreting this information. In the future, I envision integrated platforms where data from optical, electrical, and mechanical sensors are combined into a single, comprehensive dataset. Advanced algorithms will analyze this data in real time to optimize flow conditions, predict potential issues, and even provide insights into cellular function and disease states. The integration of cloud computing and big data platforms will enable researchers to store, share, and analyze large datasets collaboratively, further accelerating discovery. Ultimately, the evolution of data analytics in this field will not only enhance the performance of individual devices, but will also drive a deeper understanding of cellular heterogeneity and pave the way for truly personalized medicine.
Let's talk about the regulatory landscape. What do you see as the key regulatory hurdles for translating hydrodynamic focusing-based single-cell analysis platforms from the laboratory to the clinic, and how can these challenges be overcome?
Regulatory hurdles for microfluidic single-cell analysis platforms are indeed significant. Since these devices operate at a scale and under conditions that are quite different from traditional diagnostic tools, existing regulatory frameworks often need to be rethought. One key challenge is ensuring that the devices provide consistent, reproducible results across different settings and cell types. This requires the establishment of standardized testing protocols and quality control measures that can be validated by regulatory agencies. Additionally, there is a need for clear guidelines on data security, patient privacy, and the ethical use of genetic information obtained through single-cell analysis. To overcome these hurdles, close collaboration between researchers, manufacturers, and regulatory bodies is essential. By engaging early in the development process and conducting rigorous clinical validation studies, the research community can help shape regulatory standards that not only ensure safety and efficacy, but also promote innovation. Establishing a transparent and predictable regulatory pathway is critical for building public trust and facilitating the widespread adoption of these advanced technologies in clinical settings.
From an economic standpoint, what factors do you believe will drive the commercial success of hydrodynamic focusing platforms for single-cell analysis, and what challenges remain in making these technologies cost-effective for widespread use?
Economic success in this field will be driven by several key factors. First, the ability of hydrodynamic focusing platforms to dramatically improve the accuracy and throughput of single-cell analysis offers significant cost savings in research and clinical diagnostics. High-resolution data obtained from individual cells can lead to more precise diagnostics and targeted therapies, which in turn reduce overall healthcare costs. Second, as manufacturing techniques mature and economies of scale are achieved, the cost per device is expected to decrease, making these platforms more accessible to a wider range of laboratories and clinics. However, challenges remain in terms of standardization and reproducibility, which are essential for large-scale production. Additionally, integrating advanced sensors and control systems into a cost-effective, user-friendly package is a complex engineering task that requires further innovation. Addressing these challenges through continued interdisciplinary research and collaboration with industry partners will be critical for ensuring that hydrodynamic focusing platforms become a commercially viable solution in the competitive landscape of biomedical diagnostics.
Now, let's consider the educational impact. How are academic institutions preparing the next generation of scientists and engineers to work in this interdisciplinary field? And what skills do you think are most critical for future researchers in microfluidic single-cell analysis?
Academic institutions are increasingly recognizing the interdisciplinary nature of microfluidic single-cell analysis and are adapting their curricula accordingly. Programs now emphasize a blend of fluid dynamics, materials science, cell biology, and data analytics to provide students with a comprehensive skill set. Hands-on laboratory experiences, coupled with collaborative projects that span multiple disciplines, are essential for equipping future researchers with the practical and theoretical knowledge needed to innovate in this field. Critical skills include proficiency in microfabrication techniques, computational modeling, and the use of advanced data analysis tools. Furthermore, internships and cooperative education programs with industry partners are invaluable in providing real-world experience. By fostering a culture of interdisciplinary collaboration and continuous learning, academic institutions are ensuring that the next generation is well prepared to tackle the complex challenges and drive future breakthroughs in single-cell analysis.
As we look toward the future, what do you envision as the next major breakthroughs or applications in hydrodynamic focusing for single-cell analysis? How might these innovations reshape our approach to diagnostics and personalized medicine over the next decade?
The potential for future breakthroughs in hydrodynamic focusing is truly exciting. One major area of innovation is the development of fully integrated lab-on-a-chip devices that combine cell alignment, sorting, lysis, nucleic acid extraction, and even on-chip amplification into a single automated platform. Such devices could revolutionize point-of-care diagnostics by providing rapid, high-throughput analysis of single cells in a compact and user-friendly format. Imagine a portable device that can analyze a patient's blood sample in real time, generating comprehensive genetic and proteomic profiles that inform immediate treatment decisions. This is the promise of next-generation single-cell analysis technologies. Another transformative application is in precision oncology. By harnessing hydrodynamic focusing to isolate and analyze individual tumor cells, researchers can gain insights into the genetic diversity within a tumor, including the detection of rare drug-resistant subpopulations. This detailed information can be used to design targeted therapies that are tailored to the specific genetic makeup of a patient's cancer, leading to more effective treatments and improved patient outcomes. As our understanding of tumor heterogeneity grows, these techniques will become indispensable in developing personalized therapeutic strategies. Additionally, advancements in hydrodynamic focusing are expected to impact immunotherapy. The immune system comprises a diverse array of cells, and the ability to isolate and analyze individual immune cells with high precision can provide critical insights into immune responses. This can enable the development of personalized immunotherapies that are tailored to an individual's unique immune profile, potentially leading to more effective treatments for a range of diseases, from autoimmune disorders to infectious diseases. The convergence of advanced microfluidics with single-cell analysis is poised to transform our approach to understanding and modulating the immune system. Looking further ahead, the integration of hydrodynamic focusing with digital technologies is set to create a new era of smart microfluidic systems. As we embed more sensors and develop more sophisticated control algorithms, these platforms will become increasingly autonomous, capable of self-optimization through machine learning. The data generated by these systems will not only enhance our understanding of cellular behavior, but will also drive innovations in adaptive control and real-time diagnostics. This convergence of microfluidics, data analytics, and artificial intelligence will open up new avenues for research and clinical applications that we can only begin to imagine today.
Of course, these advancements do not come without challenges. What do you see as the most critical hurdles that remain in this field, and what strategies are researchers employing to overcome them?
One of the most significant challenges is ensuring consistent cell alignment across different cell types and under varying experimental conditions. Cellular heterogeneity poses a major obstacle, as variations in size, shape, and mechanical properties can affect how cells respond to hydrodynamic forces. To overcome this, researchers are developing adaptive microfluidic systems that can adjust flow parameters in real time based on feedback from integrated sensors. Another challenge is minimizing shear stress to prevent cell damage while still achieving precise alignment. This requires a delicate balance, and researchers are continuously optimizing channel geometries and flow rates to maintain cell viability. Furthermore, scaling up these technologies for commercial production involves addressing issues related to reproducibility and standardization. Collaborative efforts between academia and industry are focused on developing robust manufacturing protocols that can deliver consistent performance across large numbers of devices. These challenges are being tackled through a combination of advanced simulation, innovative design, and interdisciplinary collaboration.
Lastly, as we conclude our conversation, could you share your thoughts on the long-term societal and clinical implications of hydrodynamic focusing for single-cell analysis? How do you envision these technologies shaping the future of personalized medicine and healthcare?
The long-term implications of hydrodynamic focusing for single-cell analysis are truly profound. As these technologies mature, they will enable clinicians to obtain highly detailed, individualized profiles of patient cells, which is the cornerstone of personalized medicine. This will lead to more accurate diagnoses, more effective treatments, and ultimately better patient outcomes. On a societal level, the democratization of advanced diagnostic tools, especially in low-serve settings, has the potential to reduce healthcare disparities and improve overall public health. Additionally, the environmental benefits of microfluidic systems, such as reduced waste and lower resource consumption, contribute to a more sustainable approach to medical technology. I envision a future where lab-on-a-chip devices powered by hydrodynamic focusing become a routine part of clinical practice, enabling real-time point-of-care diagnostics that empower both patients and healthcare providers. This transformative impact will extend beyond medicine, influencing areas such as environmental monitoring and industrial biotechnology, and paving the way for a more connected, efficient, and personalized world.
Thank you for joining this extended discussion on advanced integration, regulatory and economic considerations, and future research directions in hydrodynamic focusing for single-cell analysis on a chip. I hope our conversation has provided you with deep insights into the challenges and opportunities that lie ahead, as well as a clear vision of how this technology will shape the future of diagnostics and personalized medicine.
This concludes our in-depth conversation on hydrodynamic focusing for single-cell analysis on a chip. We have explored advanced sensor integration, adaptive control strategies, computational modeling, economic and regulatory challenges, interdisciplinary research, and visionary future applications that promise to transform the landscape of single-cell analysis. I trust that our dialogue has not only deepened your understanding, but also ignited new ideas about the potential of these innovative systems to revolutionize healthcare and research.
Thank you for listening, and I encourage you to continue following the latest developments in this exciting field. The future of single-cell analysis is being redefined at the micro and nano scales, and your curiosity and engagement are crucial to driving these innovations forward. Let us continue to explore, innovate, and challenge the boundaries of science together. Until next time, keep pushing the limits of what is possible.